Good. Also, welcome from my side, and thank you for the nice introduction, and welcome to my presentation, in which I will talk about our work towards the integration of a human stem cell-based embryoid body model into a multi-tissue platform that we want to use for embryotoxicity testing. To give you a short introduction into developmental toxicity testing, how it is done at the moment, is that it's mostly conducted on animals in vivo. And this requires around 70% of the total animal use of toxicology studies, which is obviously quite a lot, and is of course expensive, laborious, and time-consuming, and also causes many ethical concerns, which altogether shows us that there is a clear need for novel and, most importantly, more predictive in vitro systems that can maybe at some point reduce or even replace animal studies in preclinical research. One of the current drawbacks, or one of the important current drawbacks of developmental toxicity studies, is that they are not really considering the physiological complexity of the human organism, but rather look at embryonic development in an isolated manner. However, when we look at these pregnant women here, the embryo is actually not at all isolated, but tightly connected to the maternal metabolism through the placental barrier. And the placental barrier is actually one of the most species-specific organs, and also really essential for the healthy embryonic development and growth, as it supplies oxygen and nutrients to the embryo, but also removes CO2 and waste products. The placenta also overtakes many essential metabolic, endocrine, and immunological functions, and of course, it's an important protection against toxicants and pathogens. However, nowadays we also know that not all toxicants are held back by the placenta, but that some toxicants can either just freely pass through the placenta and exert a direct toxicity effect on the embryo. Other toxicants are indeed held back by the placental tissue, but still cause indirect toxicity effects. For example, through the release of secondary mediators in the placental tissue or through the generation of metabolites. And to really evaluate both of these toxicity effects, so direct and indirect toxicity effects, it is really important to have a direct co-culture of placental tissue and embryonic tissue in the same medium environment. And this is why our idea was to model this maternal-fetal interface in vitro by combining a model of the placental barrier and an embryoid body, which is a 3D model for early embryonic development, in the same microfluidic device. And therefore, we cultured placental trophoblast cells on top of a semi-permeable membrane, which separates a maternal from an embryonic compartment. And in the embryonic compartment, we are culturing the EB as model for early embryonic development. In a proof of concept study that we published in 2021, we, however, formed the EBs from mouse embryonic stem cells, and now we optimized and revised also the entire system to increase its physiological relevance and move towards a human iPSC-derived embryoid body model that we now want to use in that platform. And in the following talk now, I would like to focus on this new platform that we developed and towards the integration of this human-based embryo model. Yeah, this is how the platform looks like. It's a hanging drop network. It is designed in a microscopy slide format, and the hanging drops are formed on a PDMS substrate, and they are connected through microchannels and thereby form an open microfluidic network. The chip is composed of two parts, a maternal and an embryonic part, which are separated through this semi-permeable membrane that I mentioned before, that is really distinguishing between the maternal fluidic compartment and the embryonic fluidic compartment, as you can also see here in this cross-section. And what you can also see here is that the placental cells are cultured on the maternal side, on top of the semi-permeable membrane, which is accessible through two lateral hanging drops, whereas the EB, the human EB, is cultured in the embryonic drop on the other side of the membrane. And one of these chips contains 6 of such units that are spaced in the distance of a 96 well plate. Yeah, we designed the entire microfluidic assay in a way that we first formed the placental barrier in our microfluidic device before we established the co-culture. That means we first seeded the placental trophoblast cells into the maternal compartment of the chip. We flip the chip upside down to a hanging drop configuration so that the cells can sediment and adhere to the semi-permeable membrane and form a tight placental barrier within the first 3 days of the assay. Then at day 3, we start the co-culture by transferring the preformed EBs into the embryonic drop of the chip. Again, we flip the chip upside down to a hanging drop configuration and start gravity-driven flow to really induce inter-tissue communication and exchange of metabolites and other molecules. And then at day 6, we can disassemble the chip again and harvest both tissue types to use it for downstream analysis. So far, on the placental side, we established an imaging-based readout, whereas on the EB side, we also established an imaging-based readout to evaluate their differentiation potential into the 3-germ layer. That means we let them differentiate spontaneously, so we don't direct them in a specific direction at day 6 and day 12 of the assay, as well as a qPCR-based readout. So in a first step, as I just mentioned, we focused on the formation of the placental barrier in the microfluidic device on top of the semi-permeable membrane here. And therefore, we used BVB30 cells, that's a placental trophoblast cell line, that formed a confluent and tight placental barrier within the first 3 days of culture, as you can see here by the cytoskeleton and the tight junction staining. And this result was also confirmed by a permeability assay for which we used a 40 kilodalton dextrain molecule, where you can see a significant difference in fluorescent intensity between the maternal and the embryonic compartment, which also shows us that we have a tight barrier in the device after 3 days of culture. In the next step, then, we focused on the establishment of the placenta EB co-culture. Also here, I first want to focus on the placental tissue under co-culture conditions, which remained its tight and confluent properties also in co-culture with the HEBs. And also that was again confirmed through a permeability assay, where we saw that the barrier tightness was not compromised by the co-culture condition. The EBs, on the other side, we preformed them in a hanging drop well plate, so under static conditions, and only transferred them into the microfluidic device after 3 days to establish the co-culture. First, we always confirmed the pluripotent nature of the iPS cells to really know that we can differentiate them into all the 3-germ layer by staining for 2 pluripotency marker TRA160 and OCT4 before the formation of the EBs. And also when we looked at the morphology of the EBs under co-culture conditions, they showed a healthy and compact morphology. However, here we wanted to go a little bit more into detail to really assess the potential of the EBs to differentiate into the 3-germ layer, to get a bit more insight into the embryotoxicity effects of drugs or other molecules that you want to test. And for that, we established an imaging-based readout first, for which we had to test different clearing methods to really get optical access through the entire 3D tissue. And here we found that the advanced cubic protocol, which is a hydrophilic clearing method, worked best for our EBs. So this hydrophilic clearing method is based on the hyperhydration and delipidation of urea to clear the tissue samples in 4 consecutive steps of fixation, delipidation, post-fixation, and RI matching. And as you can see, the EBs really became transparent in contrast to the first fixation step, and also allowed for imaging through the entire tissue volume, as you can see here on the right side, without compromising tissue morphology or cellular composition. And here I really want to mention that the hydrophilic clearing method worked best for us, also in comparison to other clearing methods, such as a hydrogel-based clearing or hydrophobic clearing method, where we saw a massive deformation of the EBs and also swelling, which of course is not really suitable for the assay. So here I really want to highlight that the clearing method you select is quite crucial for the tissue. Then we took this protocol that we developed and used it to evaluate the potential of the EBs to differentiate into the 3 germ layers under co-culture conditions, so together with the placental barrier. Therefore, we let the EBs differentiate for another 6 days, so until day 12, basically to give them a bit more time to differentiate. As we are using human cells and not mouse cells anymore, they take a little bit longer. And we also let them spread out on the surface of a well plate so that oxygen and nutrients can really reach the core of the tissue. And then we stained for 2 markers of each germ layer, so ectoderm, mesoderm, and endoderm, and we indeed saw that the EBs were able to differentiate into all the 3 germ layers under co-culture conditions, so together with the placental barrier. Then we also established another readout, so a qPCR-based readout, in which we looked at 80 different genes, so the gene expression within the HEBs. So again, classified by germ layer, ectoderm, endoderm, mesoderm, as well as some genes from the mesendoderm and pluripotency genes, to basically get a little bit more holistic picture of the differentiation potential of the EBs. This differentiation panel that we established was based on the TechMan HPSC scorecard. And basically what we did is that we took the RNA from HIPSC, so at day 0 of the assay, as well as from the HEBs at day 6 and day 12 of the assay. We created the cDNA sequence and used it for simultaneous qPCR of all the 80 genes. So basically we almost needed 1 well plate to analyze 1 EB. And here you can see the differentiation, so of the gene expression of the EBs into the different germ layer again, as well as mesendoderm and pluripotency marker at day 0, day 6, and day 12 of the assay, where blue indicates a very low gene expression and high indicates highly expressed genes. And also here we saw that the EBs were able to differentiate into all the 3 germ layer under co-culture conditions. However, we also saw a very high gene expression still in the pluripotency genes, which also is probably again due to the fact that we use human cells now that just take much longer to differentiate compared to the mouse cells. And we also observed a very high expression of ectodermal genes, which is the outermost germ layer and also the first germ layer to develop during embryonic development. So this might be the explanation here. One other thing I want to mention is that we also found out that we need really many EBs to really to see significant differences between different conditions, because, as I said before, we let them differentiate spontaneously. So we don't direct them in a specific direction, but we just let them differentiate into all the germ layers. And as the differentiation is very heterogeneous, we really need a reasonable number. So I'm talking about around maybe 15 EBs to really see significant differences between the different conditions. Yeah. So with this, I want to conclude the talk in which I presented the direct co-culture of HIPSC-derived EBs and the placental barrier, with which we aim to increase the physiological relevance of the system and of, in general, a bit of developmental toxicity test systems. We tried to have a simple platform that is pump and tubing free, also again in the context of this very heterogeneous differentiation of the EBs. And another advantage I want to mention again also is that we can recuperate all the tissues after the assay. So basically to do a wide variety of downstream analysis on both of the tissue types. And yes, the next step would then be to use the system and really study direct as well as indirect embryotoxicity effects. However, I also want to draw your attention to another talk tomorrow by Manon, which is at 12 in the afternoon in the session addressing reproduction endocrinology with MPS. And Manon will basically talk about the same chip that I just presented to you now, but she was focusing on the further development of the placental barrier. So to improve the BIVO cells, so the cell line that we used so far, by replacing it with primary trophoblast cells and endothelial cells. So, and then the idea in the end is that we combine both of our approaches and have an even more physiologically relevant system in the future. And of course, I want to thank everybody who was involved in the project, so especially the entire bioengineering lab at ETH Zurich in Basel, and also in particular Isabel, who was a master student in our lab and who was doing many of the experiments that I just presented now. Thank you, and I'm open for questions.
Oh, wow, we have time. Who was first? Okay.
Brave enough to be first. Will Lee from University of North Texas. Great talk. I have two questions. The number one question is, have you tried the development of neurotoxic compounds with your chip system?
Excuse me, could you repeat? I couldn't understand.
Could you tell me whether you have tried testing any development of neurotoxic compounds with your systems?
So far we didn't, as we started to do some compound tests, but as I mentioned before, as we need that many EBs, we didn't present the results yet, so they are not, but no neurotoxicity compounds, no.
Yeah, the second question is, why you transfer your these EBs to the well plate from day 6? Is there any reason why you transfer to those well plate?
Yes, I mean on the one side is because the placenta cells are still from a cancer cell line, so that's and they start overgrowing in the chip at some point. That's why we limited this co-culture window basically to 3 days on the chip, and also because most of the crucial developmental phases that we wanted to look at are within that window. And the only reason that we transfer it to the well plate afterwards is to give them more time to differentiate in the different germ layer. But if there was an effect of an embryotoxic compound before, we would also see that.
So then, what time point are you planning to apply your DNT compounds to your cells?
Between the day 3 and day 6, so during the co-culture phase.
Then if that's the case, the time is a little too short, because you don't see any developmental effect if you expose it for 3 days.
I mean, we tested it, we did see some effects. Yeah.
Yeah, we can take it offline probably, because we have a lot of hands. You were also, right?
I have 3 very brief questions. What is the iPSC cell line that you're using?
From Gipco, Thermo Fisher.
Name?
I would need to look it up in detail.
Right.
I can tell you later.
Second, do you see some impact of shear stress on the BO cells and their behavior?
No, because we use gravity-driven flow, so the shear stress is very low in the device. So it doesn't have a significant influence on the BO cells.
Of the flowing liquid over the BO cells?
Yeah. It's a very low shear stress because we only tilt the device by 5 degrees. So basically the shear stress is negligible.
And the third question is, you call this embryoid bodies. Can you differentiate it to gastruloids, for example?
Excuse me, I couldn't understand.
Could you differentiate your embryoid bodies to gastruloids? What would be the difference, or why do you call it embryoid body in that case?
Yeah, so we also did some tests on gastruloids. However, the gastruloids is a completely different differentiation protocol. So they really also show the spatial differentiation pattern. So they start to elongate within the first few days of culture. And the HEBs is more spontaneous, so there's not really a spatial patterning of the different germ layer within the tissue. That's the main difference.
Can I? Thank you for great talk. And I'm wondering if you observe any asymmetry condition in EB side. Then because of the placenta, there's a communication between the placenta to EB, right? So I wonder if there's any asymmetry formation, placenta side and the non-placenta side.
I couldn't understand, sorry.
Okay. So any asymmetry EB condition, like asymmetry formation in EB.
You mean a connection between the placenta and
No, the formation, any differentiation, the localization in EB or not?
Whether you have a different regions of differentiation in your EBs, like asymmetric differentiation.
No, exactly. No, so it's spatially not real. No, we don't see any regional differentiation in the EB. Yeah.
Okay, please excuse me, I just would like to make a more general comment. I developed 30 years or 25 years ago the so-called embryonic stem cell test. It is even a nature protocol, and I think for scientific fairness, you should at least mention that, because the hanging drop culture is just what we usually do in the so-called embryonic stem cell test, and the only addition that you have at the moment is a cell layer, but it's very different from, and I think you should talk to MDs or developmental biologists. You have a layer, but placenta is something very, very different, very far away. The first 6 days, if some cells on top of those, is very different from what the placenta usually does. So my point just is, it has been tested with these embryos the way you do it. They differentiate the embryoid bodies from stem cells, human stem cells, and others into tissues. It has been done, and I think for scientific fairness, you should mention that.
Thank you. I think we should move forward. Thank you, Julia, very much for your talk.